Hello, thank you for taking the time to visit my poster. My name is Julian Arrieta, and my project this summer focused on the evaluation of a new nanoparticle profiling device designed to progress early disease detection. For a bit of background, exosomes are biomaterials created by every living cell in every living organism. They range from 20 to 100 nanometers in size, and in humans play important roles in several bodily functions, such as in hormone regulation, intracellular communication, and disease response. Currently, the detection of exosomes made by cancer cells is being investigated as a new potential way to detect cancer early. Existing nanoparticle profiling devices are highly accurate, but aren't always ideal when working with specifically biological samples. They often require difficult and long sample filtrations, are difficult to maintain sterility with, or provide limited data on biological samples. The EX400 is a novel nanoparticle profiling device that can measure both hydrodynamic diameter and electrophoretic motion of nanoparticles in heterogeneous solution. This means that two important biomaterial properties can be measured with the same machine, all while requiring less filtration. Because the EX400 is still currently in development, its capabilities still need to be tested. This project aims to compare the EX400's diameter sizing accuracy against similar devices. We chose to compare the EX400 to Zetasizer's NanoZS and Nanosight's LM10, two devices often used in exosome research that use two different methods to measure nanoparticle size. To determine each device's accuracy, we measured the size of various particle size standard solutions. Particle size standard is easy to work with and is the same size every time. Here's a look at the different fluid platforms we used in each machine. And this is the resulting data. We measured a wide range of nanoparticle diameters and found that the EX400 and Nanosight LM10 struggle near the low end. The Zetasizer NanoZS performed the best across all nanoparticle sizes. Here's just the EX400's data and its linear regression compared to the actual sample diameter. Notably, the linear regression has an R-squared value very close to 1, meaning that the EX400 will reliably give the same output for a given input. So the EX400 might struggle near the low end because of how dark field microscopy works and how it calculates particle diameter. Higher brightness is needed to see smaller particles, which might expose imperfections in the fluid platform. The EX400 software could be picking up these imperfections and calculating them as large particles, causing this overestimation. Because of the EX400's high R-squared value, we might be able to calibrate its output and get a more accurate measurement. Here's a look at what that might look like. We can see that after applying this equation, the EX400's calibrated data gives us a really good estimate of particle diameter. Overall, we found that the EX400 has a lower limit in the 25 nanometer range and gives a consistent, though overestimated, output for each given input. After calibrating its outputs, the EX400 is able to measure nanoparticle size with high accuracy. Though more work is needed, this machine has the potential to make exosome research easier and help pave the way for new methods of early disease diagnosis. Thank you for your time.